Alrighty, while the rest of folks stream in, I'll start our typical announcements here. Hi everyone, good morning, good afternoon, or good evening, wherever you may be. Welcome to now the 11th week in this iteration of the Long-Term Animal Research Seminar Series. This week we hear from Charli Davies and Dr. Marcela Benitez. We'll hear from Charli first, and before I introduce her, I'll make a couple of announcements. First, if you're participating via Zoom, you should see a Q&A tab on the top or the bottom of your screen. If you open that tab, you'll be able to type any questions that you have, as well as see and upvote other people's questions. And when we get to the end of the talks, we'll go through those questions, starting with the ones with the most votes. However, if you have a clarifying question that you feel like needs to be addressed during a talk in order for you to understand something, you can type clarification in capital letters at the start of your question. And Charli and Marcela have both said that they'll do their best to answer those questions in real time. Lastly, we're going to post a recording of Charli's talk on YouTube right after these talks conclude. We're not going to post Marcela's talk, but we're going to have a recording of it. And so if you would like to watch it again, or if you know other people who were unable to attend this live, or if you're now watching this asynchronously and you're watching Charli's talk and thinking, gosh, I sure would like to see Marcela's, you can contact me either by email or on Twitter, and we can share that recording on an individual basis. Okay, so now we'll start by introducing Charli. Charli Davies is a PhD candidate in biomolecular science at the University of East Anglia. She previously completed her bachelor's degree at the University of Bristol and her master's in biodiversity and conservation at the University of Leeds. And for several years before starting her PhD, she worked with Christine Dre here at Duke, first as a field project manager at the Kalahari Meerkat Project in South Africa, and then later as a lab coordinator here in Durham. In her PhD, Charli studies the Seychelles warblers, investigating how different mechanisms interact to maintain genetic variation at immune genes within an isolated island population. Her current work focuses on understanding how pathogens influence the maintenance of MHC variation. And so for long-term viewers out there, you'll note that this is the first really detailed dive into a gene level question in this series of talks, which I'm really excited for. So I'm glad to welcome Charli here and to hear about her work. I'll turn things over to you now.
Thank you very much. And thanks both Matt and Liz for organising these talks. Let me just share my screen. Oh, it's...
So that looks good to us. Just so
yeah.
Mm-hmm.
Okay. Sorry, let me try that again. Okay, can you see that now?
Looks terrific.
Great. Yeah, so today I'll be talking to you about one of my PhD chapters. And that's looking at one immune gene, TLR3, and how this sort of evolves over 25 years in the Seychelles warbler. So first, so genetic variation is key to both the fitness and persistence of populations. And this is particularly true for small populations, where decreasing genetic variation where decreasing population size causes decreasing genetic variation. And this can result in increased inbreeding depression and then decreased fitness and loss of adaptive potential. And a number of evolutionary forces can act both separately and together to shape genetic variation. Of these, balancing selection is particularly important, as this can actively maintain genetic variation within a population. So to find out if balancing selection operates, to maintain genetic variation, it's really important to first know what and if variation is being maintained and the selective pressure behind this, and then also how and in what way selection is driving this change. So one really strong driver of evolutionary change are pathogens. So because pathogens have very big detrimental impacts on hosts have evolved various immune genes, and these are involved in the detection of pathogens. Therefore, they're key candidates in which to investigate the evolutionary forces underlying maintenance of genetic variation. So one of these immune genes, and the one I'm going to focus on today, the toll-like receptor or TLR genes. So these are innate immune receptor genes, and each TLR gene detects a specific conserved structure that's really important for the pathogen's survival. For example, flagellin on bacteria or dsDNA on viruses. So therefore, different TLR genes can detect different pathogens. As well as this, within TLR genes, various specific polymorphisms have been associated with differential host immunity. And several studies have also found evidence of balancing as well as purifying and positive selection acting on these genes. However, a few studies have gone beyond this to investigate if selection is currently acting on these genes and also what consequence this has on the population. So this brings me to the Seychelles warbler. So the Seychelles warbler is a small insectivorous passerine endemic to the Seychelles archipelago. They're long-lived species, so they can live until 19 years of age, but they have a median lifespan of 5 years post-fledgling. They were originally found across the archipelago, but in the 1960s, they went through a bottleneck, primarily due to habitat loss, and only 26 individuals were left on Kuzan Island. Since then, they've now recovered, and 4 successful translocations have taken place to 4 different islands. So our main study population is on Kuzan Island. So this is a small island with a carrying capacity of about 320 birds in 110 territories. And each bird will typically lay 1 egg per clutch. So why am I interested in the Seychelles warbler? So they're great for asking evolutionary questions for several different reasons. So firstly, as Kuzan Island is a closed population, all individuals can be followed throughout their lives from birth till death. And so we can get really accurate survival and reproductive success estimates. As you can probably guess from this seminar, it's a long-term project. So monitoring has been underway since the late 1980s. And from 97 onwards, this has increased in intensity, with over 96% of the population ringed and sampled since then. So we have really detailed information on pretty much all the individuals in the population. Specifically for my questions, because they went through a bottleneck, this has resulted in decreased genetic variation throughout the genome. So this makes them more tractable for evolutionary questions as they're not overly complex. However, some immunogenetic variation has been maintained. And specifically, there's a previous study found signature of past positive selection at 1 TLR3 SNP or genetic variant. So TLR3 is a viral sensing TLR. The genetic variant we're interested in is functionally important. So it's non-synonymous, so it causes a change in the amino acid. And this change occurs within the receptor region. And it also occurs at a reasonably high frequency. However, we don't yet know, so although we know a selective event happened in the past, we don't know if selection is currently acting on this variant in this population. And we also want to understand the functional importance of this genetic variant. So my 2 main aims today. So first, to determine the frequency of this variant or SNP and how does this change over a 25-year period in the closed population. Following on from this, I will then talk about whether anything could be maintaining variation in this population. So testing for associations between TLR3 genotype and fitness, specifically survival and reproduction. So to do this, we visit Kuzan Island twice a year and conduct a census to get biannual survival data. We also capture birds, ring them, and take blood samples. And this we can use for general genotyping and also pedigree assignment. So for TLR genotyping, we use single SNP assay, And this basically results in individuals being assigned either a AA genotype, so they're homozygous for the A allele in dark green, CC genotype, so homozygous for the minor C allele in yellow, or they'll be heterozygous AC genotypes. So they'll have one allele of each, and these guys will be in light green throughout the talk. And then, so to answer my first aim, I used just under 1,200 individuals from 15 cohorts across a 25-year period from Kuzan. And then I also used an additional 450 individuals from the 4 other islands. And for the second question, so the fitness data, we used the middle two cohorts for which we had complete data for. So onto the results. Do we find allelic change over time on Kuzan? And yes, we do. So you can see in yellow, the C allele has decreased in frequency from 39% in the population to 28% over a 25-year period. And this pattern is consistent across the different islands, although at slightly different rates. So in all of these islands, the C allele is decreasing over time. So does TLR3 variation change over time? Yes, but why? What could be driving this change? So firstly, we wanted to see if there are any associations between survival and TLR3 genotype. And we find that CC individuals in yellow of both sexes have reduced survival probability compared to AA or AC genotypes. And this results in at least a 50% reduction in median age of death from 2 or 2.5 years to 1 year. So this is quite a big change. And this equates to a 37% increase in relative risk of death for individuals homozygous at the CC allele. And interestingly, this biggest drop seems to happen in the first year of life. And in the Seychelles warbler, where they don't really have much predation, mortality is usually greatest in the first year of life. So then we moved on to reproductive rate. So we investigated the effect of TLR3 genotype on reproduction for individuals who survived past adulthood. And this is also controlling for longevity. And we did this separately for each sex. So first, we find no significant differences in female reproduction. But we did see that there was significant differences in males. So the heterozygous AC males have reduced reproductive rate compared to AA males. And when you sort of combine this survival and reproductive rate differences, we again see no significant effect of female TLR genotype on lifetime reproductive success. But this time we see that AC and CC males have significantly reduced lifetime reproductive success compared to AA alleles. So those individuals who don't have a copy of the C allele. And this is quite a big difference. So AA males end up producing roughly half the amount of offspring over their lifetime compared to AA males. So can selection be driving the temporal change at TLR3 genotype? Yes, so we see both survival differences, and in males but not females, we see reproductive differences as well. So in summary, we find evidence of selection occurring at one TLR3 SNP or genetic variant. So we see a temporal change with the C allele decreasing in frequency across populations and within a population. We also see differential survival associated with the homozygous CC individuals. And this survival difference suggests that this SNP is functionally important in this species. We see differential reproductive success in males, but not females. And this could be occurring either through mate choice or intrasexual competition. And these are both sort of key points that we're hoping to follow up. But I guess the biggest question still remaining is we see these sort of fitness relationships to this TLR3 locus, but we don't yet know what the selective agent is. We don't see any obvious signs of viruses such as pox in the population, but we haven't yet done any in-depth viral screening. So this would be another really exciting thing to follow up in the future. So overall, it's possible that the current positive selection we see might be part of a longer-term pattern of balancing selection. And this could be linked to either rare allele advantage or fluctuating selection. But we'll only be able to find this out by either monitoring further in the future or by monitoring further in the past using museum specimens. So with that, I'd like to thank all my funders and also David, Hannah and Martin, my supervisors. But most importantly, the 90+ people who have worked on the Warbler project, collecting data, analysing or genotyping. So yeah, thank you. And any questions?
I think let's start with one from Liz.
Yep.
Cool, thanks. Great talk. I was curious if you could speculate a bit on what you think drives the different rate of change for the alleles in the different islands or different populations.
Yeah, so each of these islands, although they're all in the Seychelles, they have sort of slightly different environments, but also different species of both sort of plants, insects, and other birds on these islands. So it is quite hard to say without knowing the sort of selection occurring, but you do often see differences in either prevalence or intensity of pathogens. Hope that answered your question.
Yeah, that's kind of what I was thinking. Thank you.
And then maybe let's do one more. We have a question from Mark Halber, who asks if the metrics of lifetime fitness include kin selective or inclusive fitness, or if it's just, I guess, offspring produced.
Nope, so it's just offspring produced. So both of them, we've counted any offspring that survived to at least 3 months of age to sort of try and separate the fitness associations of the offspring and TLR3 genotype and the host. So it just includes offspring, number of offspring in there. But sort of heterozygosity and various other variables are also incorporated in the models.
Great. Well, thanks very much. I enjoyed that a lot, and we'll have to hear in 25 years whether it looks like the allele frequency is rebounding and this is balancing selection.
Great.
So, great. Thanks very much. Let's see. So next, I will introduce our second speaker today, who